like that. Perfect. Okay, welcome to the first workshop of today, and it will be about Hi-C, and I would like to welcome you. Today, two people will be helping me, and this will be Sophia and Joseph, so please refer to them if you have any questions. You can raise your hand, and then you will have help from them or from me. So I also will be answering your questions. To work today, you need preferably internet access, because we will be working through Google Colab. Or if you don't have access, you will be able to run the same code on your computers if you pre-installed all the requirements. So first, let's make sure how many people have access to the internet. Can you please raise your hand if you don't have internet access, because that will be the question of the moment. Perfect. It looks like all of us will be able to work on Google Colab. So just if that's not the case, approach me in the pause or in the coffee break. We will try to resolve it. So first, what I would like you to do is to go to this link with the workshop materials. It's posted on GitHub, and here is the link. Basically, it's Genome Architecture and Function 2024 Lab. I do exactly the same, and that's the page that you should open. So it will be what we see in Hi-C workshop. Then you need to go to the IPython notebook. So it's the third file in this folder. And there you have the whole notebook that you can open in Google Colab. To do that, it's enough to press the button Open in Google Colab, or you alternatively can go to the website directly and try to open this notebook independently. I will just press this button, and here we are. Now we are able to execute the code and run it through Google Colab. Okay. So today's workshop will be consisting of two principal parts. So first, we will dive into qualitative analysis of Hi-C, and basically how to browse Hi-C data and how to learn more about 3D genomics information from that. And then we will dive more into quantitative analysis of these Hi-C matrices. To do so, let's first try to install dependencies, since we're all working on Google Colab. So for that, you will need to uncomment these lines and execute them. There will be a couple of warnings. They are all fine, and we just need to run the code. What is the software that we're going to install? Most of these tools are OpenToC tools that the community of developers in Chromatin, Open Chromosome Collective, as we call ourselves, are developing. And I will say a couple of words more about this software, including Cooler, Bioframe, and a couple of other utilities, and cool tools, of course. Okay. While the software is installing, let's first dive into Hi-C data. I hope Hi-C doesn't need particular introduction, so it's the method to probe three-dimensional organization with the next-generation sequencing methods. And the data is recently exploding. You can see it on the left. It's the screenshot from the 4DN data portal, where basically researchers from all over the world who belong to 4DN community post their data, and it's now available for a bunch of species, and the amount of the data is growing. Of course, it's also posted on different other resources such as ENCODE, GEO, and so on. So you can find a bunch of sources for Hi-C data, but let's try to explore this particular one. So I will follow 4DN data portal, and I will select some sample in situ Hi-C dataset for human. You can also do the same. You can also follow the link for 4DN. It's present in your notebooks. If you scroll a little bit below, you will be able to see the links to the public Hi-C datasets. And there you will be able to click 4DN data portal and basically follow the same actions as I do. And here at 4DN portal, I go to some sample Hi-C human data. You will see that there is a bunch of cell lines and tissue types available for human. And let's try to navigate to one of them. For example, it can be human embryonic stem cells, H1 ESC. I will follow, and you will be able to see already the representation of the data. Let's try to explore it. And here it goes. Basically, this is the Hi-C dataset, where you can see all the human chromosomes. We can try to zoom in and look at the interactions between the parts of the same chromosome. For example, here, intrachromosomal interactions of the first chromosome. Let's try to zoom in further. So now you can basically see additional information available here. It's also information about the genes. So on top, you can see different genes. And on the map itself, you will be able to see what are the features of 3D genome associated with this particular genes. So for example, what do I see? The first signal I see is the main diagonal, which is very, very bright. And that's what we expect, because regions that are located at the same, close to each other, at the same chromosome next to each other, they will be interacting the most. So that's why we see this bright diagonal. And next, we see that there are domains. So there are these squares across the main diagonal. And these are domains or topologically associating domains, stats, that are known to fold the three-dimensional genome. So these are some examples of the features that you can find in Hi-C data. And this is a nice way to see them. Basically, you can browse them right away, find the gene that you like, or find some chromosome that you're curious about, or do simple quality control of this particular data set. So this is a nice way to go, but let's try to emphasize a little bit on what is the actual engine that we need for visualizing Hi-C dataset. And actually, showing Hi-C data that I did for you is not that simple task. So it's not simply that we go from some Hi-C data deposited on the website to the image. In fact, it's much more complicated task where you need lots of software in between that will be fetching not all the data, because Hi-C data can be several hundreds of gigabytes of data. You, of course, need a small view. So the problem is that you need to go through this immense data through a bunch of software and get a small piece of your data set that you would like to show and would like to see and inspect. And that task is resolved by one trick where you need to fetch only parts of the data. So you fetch the tile set, part of the whole Hi-C matrix, and you do that at multiple levels of hierarchy. Because you can look, as we saw, we can look at the whole chromosomal level, but we can zoom in and zoom out to different scales of genomic locations. So this trick is performed by several browsers, and all of them are running on top of HiGlass. HiGlass is the engine which is resolving this type of issues for you and allows you to browse Hi-C data in rather fast and simple way. So these are the example of the tools that are based on HiGlass. We covered already 4D nucleome, but I want to emphasize here a very informative website called highlass.io, where you can find already numerous data sets preloaded for you and put in a nice way, in a nice set of examples of what data can be visualized and analyzed together with Hi-C. So this is highlass.io, where you can take a look at many different data sets. So let's zoom to some particular one. And for example, let's do this one. Here you will be able to see two data sets of human cell lines, and there will be two views with different Hi-C data. Might be a little bit slow because we are all trying to access it at the same time. So if I don't want to wait for it, this is the website for you to explore and see what different presets are available and what are the opportunities to visualize Hi-C data together with other omics or other types of data. Okay. HiGlass has advantage that it can be indeed incorporated into a website. And for example, in our lab, we also have we utilize this property. And in the third link here, you can find our lab website where we just have one project where we generated, our collaborators generated some data, we analyzed it, and now we put it as a linked view within our website, which is very handy, and you can associate useful information from the published paper together in a nice interactive view that you can browse and learn more details about the published dataset. So this I find very, very useful and helpful. So I can recommend for you to check out HiGlass and how you can utilize this. And finally, there is an interactive HiGlass-based browser where you can deposit your own data and save the views for the future. It's called resgen.io, and I recommend you not to follow this link at the moment, because if we all log in at the same time, it will be just ruined. But this is basically the Genome browser where you log in, you load your own data, you visualize it, and you store the views for the future. If you are curious, you will be able to check out more details about ResGen and basically how to work with it, and you will find useful tips for that in the presentation associated with today's workshop. If you go to GitHub and you will open a workshop cheat sheet, you will be able to find all these hints about how to work with different versions of HiGlass, including ResGen. Okay, so today, after we browsed HiC data a little bit, we would like to do that in a more dataset-specific view. So now probably on Colab, there was a warning for you that you need to restart the session, so please do that. And then go to the section 2 of first part called Interactive Browsers for HiC. There will be a section Explore HiC data in your notebook with HiGlass Python. Oops. And then you can start executing the code. Ideally, import HiGlass should work perfectly with no warnings. And then you run the next cell. Basically, you can press this button, play the cell, or you can type Shift-Enter, and it will be doing exactly the same. What you will see now in your Google Colab will be this nice HiGlass view. What is this particular dataset? So this dataset is not entirely HiC. It is the HiC version called microC, which is a more advanced version of HiC, which allows very fine-grained resolution of the 3D interaction maps. And you can try to zoom in, and you can do the same things as you do in the browsers before, but now in your view. So please take your time and try to zoom in, try to find the gene that you are working with or you are curious about, and try to find different HiC features that we were talking about. For example, you can see domains, you can see different chromosomes, intrachromosomal interactions, and also pay attention to this checkerboard pattern, which is called compartments. Note how compartments are also nicely pronounced in this dataset. The data is mouse embryonic stem cells, so this is not human anymore. And this you can see reflected in annotation. So on top of this view, you will see the genes and also annotation of the assembly. This is MM10 mouse assembly. What else can you do with this nice view? You can try to change the color scheme, because if I try to zoom in, for example, here, I cannot distinguish the features as well as I was before. So maybe I would like to adjust the color bar a little bit so that I can see that intensity of the color, dynamic range of the color has changed. So for example, if I do it like this, I do not distinguish many features within this big domain at EPHA4 gene. But if I change the color bar a little bit, I'm able to see very interesting structure at this gene, which we can call a stripe. So it's like one-sided increased number of interactions at the beginning of this gene. So this feature is usually called stripe or flame, depending on the paper. And that's what we can nicely see if we adjust this color bar. How are all these features formed? We see that there are sometimes stripes, there are sometimes domains, sometimes you can see these nice dots, or some people call them loops. So what is the mechanism responsible for them? So far we know that these are the factors of, these are the structures formed by the mechanism of loop extrusion. So there is a specialized complex of proteins called cohesin, that loads onto DNA and starts to create the loop progressively up until it meets the other protein called CTCF bound at specific locations, and then cohesin cannot progress anymore and it stalls at these CTCF sites. And this process creates this nice pattern that we can see in this IC matrix. So the CTCF sites, the stop points for cohesin, will be located usually at the stripes and at the dots in high-C matrices. And in the next task, 2.3, if you scroll a little bit below and you execute the next cell, you will be able to see two views now. I will enlarge it a little bit. And in these two views, I provided you the same data set on the left, and on the right, you will be able to see the high-C data for the cells with removed RED21. So very briefly, we can add the Genome tag for a protein of interest, in this particular case, element of cohesin, RED21. And then upon treatment of auxin, we can fast degrade this protein. And we can take a look what absence of RED21, key component of loop extrusion, how that will affect our high-C matrices. So from the first side, nothing specifically changes. So there are still chromosomes. We can still see enriched interactions within chromosomes and far less interactions between chromosomes. But let's try to zoom in some particular chromosome. It is updating rather slow, but we can take time and wait. Here it goes. We see that the views are slowly updating. And what we can notice already is that on the left, there are lots of domains and stripes, and on the right, there are far less of them. And this is the nice example when on the left we see this bright stripe, and on the right it's completely gone. You can find more examples like that, where there was some feature on the diagonal, close to the diagonal of high-C matrix. It was either stripe or dot or domain, and then it is gone upon RED21 degradation. However, not all the features are gone, and some features persist. For example, here you can see a nice example where there was a loop, but this loop persisted upon RED21 degradation. This means that this feature indeed was presumably loop extrusion independent. And interestingly, when we see this case of some structures that still persist in RED21 degradation, you can notice that they form this checkerboard pattern again. So the checkerboards are still there even without loop extrusion, which means that compartmentalization is still there and it's independent mechanism from loop extrusion. this is a nice theory that you can learn from these comparative views, but let's try to dig deeper, and let's try to see with what particular chromatin factors the structures might be associated. And here I encourage you to run the next slide, where you will see that there are two views, but there are now additional data sets on the left. I'll try to enlarge it a little bit more so that you can see. So here I added a bunch of ChIP-seq data for you, which is the method for assessing where different chromatin-bound factors are located in the genome. And I added for you the ChIP-seq for CTCF, the factor which is responsible for stop points of extrusion, and I added a bunch of different histone modifications. So it might be loading slowly, but after some time you will see that there is indeed a one-dimensional track. So for each position in the genome, there will be a value associated with each histone modification or with each track on the left, and you will be able to see where this histone modification was enriched. For example, this region that I point to is clearly enriched in K4 trimetallation, K27 acetylation, which is the activity histone mark. So please take your time while this view is loading. In my case, I see this trouble. Tile set info not found. If you encounter the same issue, please just run the same cell again, and it should resolve it. Meanwhile, I do not know whether it will be fine for Google Colab because we are all logged in. We all tried to run the same code there. So let's try to take a look at what we can get out of this view. So I will enlarge the answer part for this section, but later during the break, if the code is finally executed, you will be able to see that by yourself, and you will be able to find the gene that you like, for example. So this is one of the examples. For the same data sets, on the left, wild type, on the right, red21 degradation. And now we can look at different histone modifications and see what patterns they correspond to. So the first track, it's CTCF, and you can clearly see that there are very narrow puncture peaks in there, which corresponds to CTCF binding. And if you try to draw the line, indeed they will be corresponding to the locations of these nice high-C features, such as dots and domains. They are gone in red21 degradation, and nicely, these CTCF peaks were associated with some active histone modifications. So here, for example, it's K4 trimetallation, mark of activity, and it's nicely corresponding to the CTCF peak and to the formation of these structures in high-C. However, there are also some regions which are inactive, and these are K27 trimethylation regions, so polycol, and this is the region which is supposed to be actively repressed. And we see that it corresponds on high-C to a very little domain, and this domain persists in red21 degradation. So these polycomb bodies, as people call them, are persistent to the loop extrusion abolishment. You can see that the structure is still there. You can explore this more, and you can take a look, for example, at these nest structures that are still there despite cohesin being gone. And they nicely correspond to some activity marks. You can see that there is some active chromatin there, and it also maybe corresponds to the formation of compartments there. The structures might be independent of extrusion. If you see this trouble, that the views are a little bit asynchronous, you may notice that the bottoms of these two views are not coinciding. So you can just pull a little bit the one which is smaller, and then it will make them equal in size, and it will be much more helpful to synchronize them and view the features simultaneously. Again, it's this lower corner that you can pull a little bit and make sure that they're the same size. It's just a small tip that you can use to optimize the high glass view. Any questions or suggestions or concerns? Anybody paid attention to this upper track? What is that? This is actually rather interesting information, and for me it's an amazing way to integrate upon multiple data available. So on the left I was showing to you a bunch of different ChIP-seqs, and you need to see where are the domains of active chromatin, where are polycomb domains, and you need to analyze 5 tracks simultaneously, which is not super convenient. And to resolve this issue, people came up with the annotation of chromatin states. And in this case, it's called epilogos, one of the approaches to create these chromatin states. And this is the integrated dataset upon multiple ChIP-seq for mouse embryonic stem cells that will allow you to see what is the consensus type of chromatin at this particular location. And you can see some of the regions are just general heterochromatin or not annotated, but some of the regions might be active, transcription start sites, or enhancers. So if you hover over it, ideally it should tell you what is the type of chromatin associated with this particular region. And this is super helpful because you need to analyze much less data, you comprehend it much more easier. And this is a nice example where there is an active chromatin here, and on the right it will correspond to the compartment. It's not super visible now because the view is not updated properly, but you already can see that this big blue chunk here corresponds to the compartment formation on HiC-map. Yeah, it's very slowly updating, and I hope you can appreciate the power of this view and how easy it is to see what the structures in HiC are associated with. Moreover, if you are curious about some particular gene, you can take the name of this gene and insert it in the search box. It might not work now because we are too many users and it's very intense view. But ideally, you should be able to zoom in this particular gene location on your HiC-map and check the structures around this gene. Alternatively, you can just pick up any gene that is in the vicinity of where you were able to zoom in. This is a nice example, I believe. So this is a gene which is actively transcribed, is supposed to be actively transcribed in mouse. It has strong transcription chromatin state, and it nicely corresponds to the compartment here, which is, of course, independent of loop extrusion. That's why it's present both on the left and the right view. Now you see that I face an issue. The view has updated, and I am too much zoomed in. The resolution here is just 1 kilobase, so each pixel here is too small, and the data is too sparse to see anything reasonable. How do we resolve that? We can go to the settings of this view, and we can set up the zoom limit. So again, if I hover on the top right corner of my view, I can press the gear button, select the dataset, configure series, and then I zoom in and set up the resolution. For example, I would like to analyze it at 5 kb, which is much more reasonable to see loop extrusion features, and it won't be that sparse. And I do the same for the view on the right. Here it goes. And now you can see that it's much more easy to analyze these views, see the features, etc. Another small trick I would like to show you is the change of the scale. So here you can see that the scale is not entirely linear, it's logarithmic on the color bar. And it might be not beneficial all the time. we might try to change it to the linear scale. So again, I go to the settings of the view. I go to the value scaling, and I set up the view to linear. Now I need to, the field is blank all of a sudden because the color bar is not adjusted appropriately, and I need to change it. Here it goes. And now you can see how much more pronounced are the dots and stripes in linear view here. It's one of the tricks that you can also use for visualization of your Hi-C data, because some of the features are indeed more pronounced and more visible if you change the color scale. With that, I suggest to switch to the next section and maybe go to quantitative analysis of Hi-C maps, because we are through visualization, through our first qualitative analysis of Hi-C maps. And I hope I left you with the instrument that you might use in the future, and it might be helpful to you. For the next part, I would like to tell you a couple of words about quantitative analysis of Hi-C. So this is the fundamental problem that we faced in the lab, and people before me in the Mirny lab faced. And they wanted to develop the software that will be robustly showing information, useful information about Hi-C matrices and features in there. And they started to develop the software that will be accessible to the broader community, that will be open source and that many people can contribute to. And that gave rise to the Open Chromosome Collective. So here you can see Mirny Lab and some people who were at the beginning of the Open2C, including Maxim, who is today here and who was one of the founders of this initiative. And these are the people who are currently involved in this Open2C. We have a nice GitHub repo where you can see the tools that we develop, and we have also Twitter and Slack. And on Slack, we very much welcome any questions and comments and suggestions. So please join. I first would like to start to switch to the code and maybe try to run the first cell with the import of all the software, and then we will switch to Cooler, to one of the fundamentals of Open2C software, the instrument that stores and allows you to query Hi-C data in a very effective way. So let's go to the part 2 of the Google Colab, and let's go to the point number 1, import all the software. Here you can see a bunch of standard libraries for data analysis in Python. NumPy, Pandas, Matplotlib for visualization. And in the end, you will see Open2C software, including Cooler, Tools, and Bioframe. So that's these are the key 3 packages that we will be using today. So what data we will be working with? We will be working with the same micro-C data as we did for HiGlass Spark. And for your convenience, we already uploaded it for you. And if you type print available datasets, you will see the list of multiple Hi-C, ChIP-seq datasets, and also micro-C for mouse that we were working with today. They can be automatically downloaded. The next section here will tell you how to do that. You specify the data folder, the data folder. Yeah. You specify the data folder, and then you say CoTools download data with this particular prefix, with this particular key. So it will be mouse embryonic stem cells, DRED21, meaning that degron is attached to RED21 protein. UT means untreated. So this will be wild type phenotype of the cells. They do not have any perturbations, except modified RED21, which is supposed to work fine on the genome. So after that, you will be able to see that now in your data folder, there is mpool file with this particular name. I execute the cell, and I see that the data is downloading. It might take around a couple of minutes because it's the dataset which weighs around 1 to 200 megabytes. And today we will be comparing that to different perturbations, to RED21 IAA, which means auxin-induced degradation. So this will be the treatment. We will also see the data sets for CDCF and the WOPL degradations. So run the next cell, and it will download the remaining 3 data sets on your local Google Colab repository. At this moment, please pay attention to the file names. We will use them throughout these notebooks. So basically, that's the variable that will hold the file name for this particular treatment: CDCF1, WOPL1, RED21, and untreated UT. So we will need these 4 files. You can come back to this top of the file and this particular cell if you need the name of this particular files again later. While we are all waiting for this download, I will stop it. You cannot stop it. I just want to proceed and show you the cooler structure, and I will run the download further. You can just proceed with what you are doing right now, and just look at the screen following what I will tell you about cooler files. So we can take a look within one cooler file. So for example, this untreated cool file. Inside, you will see a bunch of datasets for different resolutions. So this is, for example, 1 KB, the smallest, the finest available resolution. And then these are other resolutions available within this file up until to 5 megabytes, pretty coarse grain datasets. They are all deposited in this data type already at the same time. Now, let's select one particular resolution. How do we load Hi-C data for that? And this is an example here. So you just ask cooler, open the file, and you specify one particular resolution after 2 double dots. And let's try to execute it. Now, CLR will be cooler object that has different attributes with chromosome names, for example, or information about the resolution or bean size. It also has the feature called beans. So basically, you can see that it's a table with information about each bean in the chromosome. Chromosome start, end, and there is some additional information about this cooler file for this particular genomic position. You can notice that not all chromosomes are present in this table. There is only 1st and 12th chromosome, and that's for simplicity, because I do not want to load the whole genome for you. We just want to run a small test. That's why we have 1st and 12th chromosome, for example. And that today we will be working only with these 2 chromosomes in the upcoming examples. Another interesting feature is CLR pixels, pixel table, and this is the key information for the Hi-C, because here you can see for each pair of genomic loci, different beans of the genome, we will have the number of interactions that were obtained from the Hi-C experiment. And interestingly, not all the pixels will be present here, and only those that have non-zero values. And this means that the data that we deposit is sparse. We do not store the whole matrix of pairwise genomic interactions. We store only those contacts that are non-zero, and that allows us to save lots of space and time. Cooler software is a sparse storage of Hi-C data. And indeed, the key components of it are different tables. table with chromosomes, table with beans, and table with pixels. There might be some metadata associated with it, but basically this is the key structure of Cooler file, and the Cooler library is just an interface for this particular structure in the file. You can read more in the documentation of the Cooler. So basically, how do we fetch a Cooler dataset? And let's try to do it later a little bit. So if I loaded a CLR file, how do I ask what particular interactions look like? And using the information from the docs, I will need to run clr.matrix, and I will just fetch some of the locations. For example, I want chromosome 1. And you can see that the output of this fetcher will be the NumPy matrix, two-dimensional matrix. And we can further use this matrix, we can visualize it or compute different features in it. Okay, I will proceed with downloading the data. Okay, I think the data is loaded. And now I'm able to progress to the first part of the quantitative part of this workshop. Let's try to visualize the data in Python. First, let's load chromosomes. You will see that there are two particular chromosomes, 1 and 12. Let's load the whole data. So I specify CLR matrix and just query all of it. So we will be loading all the data set at resolution of that was specified before. So when I was loading the data, I mentioned that it's 1 megabase resolution. So now the whole data will be loaded at this particular 1 megabase resolution here. And I visualize both available chromosome with very simple Python matplotlib functions. So it's matShow. And first I create the figure, then I visualize the dataset, IC dataset. I place color bar, I adjust the axis a little bit. And now you can see plain visualization of IC. It's good enough. You can see already the main diagonal, you can see compartments, you can see between chromosomal interactions, but we can improve it further. As the next task, it's a little bit more complicated code, but it's basically all visualization that probably you run a lot in your daily life as well. It's visualization of the part of the chromosome. So it's basically similar structure to the simple code snippet that I provided to you before. So we first create the figure with some adjustments. We query IC data. We visualize this data by matShow. And in this view on the right, you are able to see different extents at which we can look at IC data. We can look at single chromosome, or we can zoom in particular region of the chromosome. A little bit note of how we did that in the code. So when I was loading IC matrix, when I load the whole dataset, I specified matrix and load everything. So basically this double dot means load the full dataset. If you're loading your dataset at 1 KB resolution, I do not recommend using it because it will overload your memory. So do not use it without thinking. Much more wise way will be to specify the regions that you would like to fetch your IC data from. And in this case, this is one particular chromosome. And here is how I load region of the chromosome. I need to specify both start and end of the chromosome on top of that. And the syntax for loading will be very similar. How can we improve this further? You can see that the scale here is linear. we can try logarithmic scale. We know already from high-glass views that was resulting in pretty nice pictures. So now, there is a very helpful thing in matplotlib. You can specify the color scales with the intrinsic matplotlib functions deposited in the colors library. This is very helpful because you later can control what will be the limits of your color bars in a very good way. I provide you in the cheat sheet a little bit of tips on how to control the scale, and I think it's a very robust and helpful way. So please note this matplotlib colors and different types of normalizations provided in there. They provide also an excellent documentation on what is available. So basically, as you can see, in my case, I set up the maximum of my logarithmic scale to 0.1, and it's nicely reflected in the views with high-C matrices. And you can appreciate how much the view has changed, because at the previous view, I was mostly able to see the main diagonal. And in the logarithmic scale, I'm now able to distinguish different interactions between compartments, and both within chromosome and in between chromosomes. So if I look at this particular square, to this particular regions here, the compartments are pretty visible, although they were indistinguishable in the view with linear scale. So mind the color bar, mind the color scale that you use, and try to adjust it for your views for the better representation. This is the default color map called Viridis, but we can try to use something more visual. And these are two examples in the section 1.4 color maps. These are two examples of very popular color maps for high-C data. So you can see that different features are actually more visible with different color maps. This is that we call fold, or creatively it's called pumpkin spice. You can see that the compartments are more visible and the contrast close to diagonal is more comprehensible, while in the red's color map, it might be more hard to distinguish in the logarithmic scale. While in linear scale, it's not that bad. You can see that the compartments here are pretty visible and maybe even better than in the full color map. And here I give you an example of more high-resolution dataset. So I load cooler file at 10 KB resolution now. I define another variable containing cooler file with 10 KB1, and acquired much more small region on the high-C matrix. And here it goes. You can see what color map is actually more helpful and what scale, linear versus logarithmic, is more useful. In my opinion, local features are much more visible with the linear scale. You can see the dots and stripes very nicely, while logarithmic scale allows you to see domains, more broad structures. So now I've shown you the tricks how to work with high-C data in Python, and I would like you to do a task now where you will be loading two datasets at the same time, untreated and red21 degraded. And then I encourage you to pick some region and visualize these two maps simultaneously and compare against each other. Just make sure to select the region from chromosome 1 or chromosome 12, because these are two available in the datasets. In my case, I will probably select the same one as we did this example for. So I need to provide this string annotation of the chromosomal region. And I visualize it. So take your time, select the region, experiment maybe with different resolutions or different sizes, because here it's 5 megabase, and you might select something more narrow or more broad. And this is an example of output where you will be able to see the effects of Red21 degradation. Okay, and there was a nice question from the audience while we were working. So what is the difference between stripe and dot, and how loop extrusion contributes to that? And I bring to you here a nice example of the stripe with the dot nearby. So what is the difference between them, and how they can be created by loop extrusion? In this case, you can see the extruder that can be loaded on DNA up until it encounters CTCF. What happens when cohesin encounters CTCF? It stops there, and it cannot progress any further. So it will create more interactions up until CTCF, so between two CTCFs nearby. So for example, this CTCF and another CTCF in the convergent orientation. So not any CTCF will work in this case, but only those CTCF that are facing towards each other will be boundaries for cohesin. So when cohesin reaches them, it will be stopped there, and it will create a more stable loop between two CTCFs. On the Hi-C matrix, how that will look like, if this were two CTCFs, for example, one CTCF, I will plot a line here, and another CTCF over here, right? So cohesin, which loaded within, it will be progressing up until it encounters those CTCFs in the corners, and it will create a dot between these two CTCFs. The same with this dot, or with this dot. But this structure is much more interesting, because here you can see that these three dots are kind of connected with some stripe-like structure. Why it can be happening? Imagine that cohesin is loaded at some location within this domain, for example here. And then it moves with both legs, with both sides of it, it moves in both directions, and at some point one leg will encounter CTCF, for example this particular CTCF here. But another leg will be freely progressing forward. That's why we can see that this is not a single dot, but it's a prolonged stripe of interactions of cohesin which is stuck with just one side. And that's how this nice pattern can be created in Hi-C maps. And this is the key difference between dots and stripes. That dots will be those that are bound by CTCFs in two locations. Stripes are those where cohesin is stuck with only one side. With that, it's a good moment to switch to another task and think about other patterns that can be created by loop extrusion. And with this, let's try to switch to the part number 2, section 2, insulation. This is the first basic analysis that you can run on Hi-C data, trying to assess where we encounter more or less interactions around the diagonal. This is one of the definitions of the domain. If the number of interactions next to the diagonal is unreached, that will be domain, topologically associating domain or tag. And if there are less interactions, it will be the boundary. We know that usually CTCFs are associated with these boundaries because cohesin won't be able to create loops across the boundary. And we want to have a way to assess that in our Hi-C matrices. And for that, one of the traditional ways is to create insulation profile. Basically, it's a very simple approach where you take this sliding window of particular size and you calculate the number of interactions within this window, and you slide this window across the whole chromosome, and you get this normalized number of interactions. If there is a dip, it means there were less interactions. If there is a peak, it means that there were lots of interactions. And we can try to find the dips in the profile and say that these are the boundaries. You can do that with the library insulation of CoolTools. So let's go to the section number 2.1 and let's try to run the code. We will be running it for 10 KB resolution. So the size of the single pixel in Hi-C matrix will be 10 KB. We also specify the windows because it is a free parameter in our approach. Indeed, we can take this insulation window and slide it, but what should be the size? We do not have any knowledge, a priori knowledge of how windows should look like. So we take different sizes of this window and we anchor them to the resolution because it's a meaningful starting point. We have elementary bin size, and now let's try to take 3 bin sizes, 5 bin sizes, 10 bin sizes, and let's figure out which one is working the best. This step might take some time because we run the computations for all chromosomes at pretty high resolution. And here it goes. We created the insulation table. Let's take a look inside. Here it goes. So for each position on the chromosome, we get information about insulation score at a given window size. We get some additional information about the quality, number of valid pixels. If this number is large, it's good. If it's close to 0, we would like to get rid of this regions because they're low confidence. And finally, we will have the information on boundary, whether there is boundary and what is the strength of this boundary. Formally, how do we define the boundary strength? This can be done in multiple ways, but what we try to find the depth of the deep relative to the nearest highest peaks in the profile. So basically, this is the boundary strength that you can find in this table here. That's good. We calculated the table. Now let's try to make sense of it, and let's try to visualize it. I provide for you a small function how to visualize Hi-C in a nice rotated way, because you notice here we display Hi-C matrix not as usual beautiful square. It's now square put on the main diagonal. So it's kind of 45 degrees rotated Hi-C matrix. And you can also do the same in your code. So it's the function 45 degrees rotated color mesh here. So you can just execute it and use in the next cell. Here it goes. I will zoom it in. You can see an example of Hi-C region where you can already appreciate that there are some boundaries and there are different levels of strength. For example, this is a very strong boundary, it's very clean, but there are also some minor ones. For example, on the right here, clearly you can see large pronounced boundaries, but also some more minuscule tiny details of the domains. Let's look at the insulation profile, and these are different lines for different window sizes, and you can appreciate that not all window sizes are helpful. For example, the smallest window size, 30 KB, is super noisy, it's hard to interpret it, and there are lots of teeny tiny dips in this profile. If we take the largest window size, for here it's 250 kilobase pairs, it's also not that helpful because it's too smooth and it highlights too little boundaries that we can see by eye. For example, in this region, it picks up only the strongest one, and these ones are basically gone and you cannot see them anymore in this orange profile with 250 KB. We need something in between. And indeed, if we take a look at 100 KB, it's kind of much more sensible, and it creates the dips in the locations where we prefer to see the TED boundaries. So let's proceed with 100 KB. And let's take a look what boundaries were called at this resolution. Interestingly, this nice function, insulation, it pre-calculates the boundaries for you. So you don't need to do any other action. It will output you the boundary strength. Now, let's try to select window size. We decided that it should be 100 KB. And let's try to visualize the boundaries. What this code is doing, it's showing you one particular insulation profile, and it shows you the boundaries, weak, all the small dips, and also the strong ones, very pronounced dips. Quick note on how we compute strong versus weak. There are different ways to do that, but what we prefer at OpenGC is to visualize the distribution of boundary strength. And then, apparently, at some resolution, we will see that this distribution is bimodal. We have the larger peak at larger values, and then we have a second peak on the smaller values of weaker boundaries. And we can make it a cutoff, because this is the noise, and these are the actual boundaries that we are curious about. We can create the cutoff, and then select those that will be strong versus weak. There are several data processing approaches to find this cutoff in the bimodal distributions. And by default, Lee criterion is working pretty nicely, and you can use it as the separatory boundary between strong and weak boundaries. Okay, that's a nice opportunity. You now know the positions of the boundaries, what we can do with them afterwards. And here is what people usually do with this kind of information. This is a pile-up plot where we take the snippets of IC metrics, and then we average them all together to produce the average profile, average information of IC. So we can try to do the same. So let's try to take all the strong boundaries, and let's try to take the snippets of IC around the strong boundaries. So it will be the squares around each orange boundary. And let's try to average them. For that, we will use the function coolTools_pileup. So you can see that we have 977 strong boundaries in the dataset. And here it goes. Here is the average boundary, and you can see that on average you indeed see insulation on the boundaries. Any questions or suggestions? Because the next task will be to do the same, but on red 21 degraded cells, where there is no loop extrusion, and check out whether this insulation is still persistent. And just to highlight it again, that how we define insulation. Insulation is lack of interactions between neighboring loci. And you can see this insulation boundaries here as white big triangles put on the main diagonal of IC matrix. And we tried to assess these dips in the interactions with calculating insulation profile. Then we found boundaries, we found the dips in the insulation profile, and then we tried to pile up multiple locations with a strong insulation and create the average profile of wild-type mouse embryonic stem cells, how the boundaries look like there. And you can see that there is indeed average insulation there, meaning that there are lack of interactions between regions to the left and to the right from the central point. And interestingly, if you do the same in red 21, I show you the answer. Probably you were able to run the same in your code. So it's here in practice task. You will see that there are no interactions, no interaction deep in red 21 degradation. Insulation is all of a sudden gone if we remove cohesin. And it means that most of the boundaries that we detected were indeed cohesin dependent and were the feature of loop extrusion. And as I mentioned before, mechanistically, how we can arrive to this figure, we can look at mechanism of loop extrusion, and sorry for that, and we will see that cohesin is predominantly linking regions between neighboring CTCF boundaries and not across these boundaries. So the event when cohesin will hop the CTCF side, hop over the CTCF side, will be more rare, and we see that cohesins do not create interactions across CTCF boundary. So that's why we see this insulation. And if we have no cohesin, there will be no more insulation, and we will see this nice, nearly blank featureless Hi-C on average. Okay, and this was on diagonal Hi-C. So once again, I will quickly show you how it looks like in ResGen, in HiGlass, sorry. So you can see here, there is some cell type, human embryonic stem cells, and you can see this nice domains, insulation, dots, and stripes, major features of Hi-C. And for your convenience, the authors of this 4DN portal, they pre-calculated for you the profiles of insulation. So actually, on top here, you can see the insulation profiles, and you can see the boundaries pre-calculated for this particular data set. And this is super useful because now you are able to see at what particular genes the boundaries are located. You can navigate and see whether the deep is pronounced enough to be considered in strong isolation, and so on. So if you browse any data set of your interest at 4DN Data Portal, it will be nice insulation profile and boundaries pre-calculated for you. So, okay, we're done with insulation. It's a good moment to ask any questions about insulation if you had some. If not, we will progress to compartments. And here I will switch to this nice HiGlass view with human embryonic stem cells, but on a larger scale. This is almost the scale of the whole first chromosome. And there you can see this checkerboard pattern called compartments. Let's try to figure out what that is a little bit further. So let's consider one particular region of the genome. For example, let's take this big block here, and I will mark its boundaries here with vertical and horizontal lines. So you can see this domain is interacting with other regions in the genome in a preferential way. So it likes to interact with this one, for example. I will mark its boundaries. You can see there is a preferred interaction between this block of genome and this block of genome. And there is enriched interaction block at their intersection Hi-C matrix. However, the same region doesn't interact with the one right next to it. And you can see this blank square or white checkerboard pattern. Now, if we look further across this line of interactions created by this region, you will see that it, for example, likes to interact with some regions downstream more. This line, next line, and further downstream blocks. So this is exactly the preferential properties of this particular region. So it likes to interact with more regions of its type, and we usually say, if there are many genes, we usually say that it's active region, and we will notice that other regions with which it interacts will be also active. So this will be called a compartment. Now let's take a look at what we excluded from our picture, regions B. So right next to it was the region B, this one and this one, were B compartments, and they avoided interaction with A compartment, so you can see it as this big, more or less white part of interactions. And then this B compartment will be preferentially interacting with something else. So you can see that there are some block-like regions with which it still prefers to interact. For example, this one here, I will put vertical line here to mark this. Here it goes. For example, this one. So this creates the opposing pattern of the checkerboards, and how we usually calculate it, it's by calculating compartmental profile. And in HiGlass, you can also see that here. So you can see the annotation of A, B compartments on top. Yeah, I might have messed the sign of the compartments when I was explaining it to you. Usually we assume positive sign is A compartment, negative sign of this value is B compartment. So it will be nice A compartment, then B compartment, then again A and B. How was that calculated? Usually, to get this pattern of interactions and linear information, we run the procedure of eigenvector decomposition. It's a way to get more linear data from your Hi-C. Hi-C is two-dimensional matrix, and you want to have just one-dimensional track describing the pattern of interactions of compartments. So to do that, you run the procedure of eigenvector decomposition. Here I just broadly say that it's compartmental calling, but what's running under the hood is that we take Hi-C matrix and we decompose it into a nice sum of vector products. And it's a math operation that you don't need particularly to well understand, but the important part is that if you take the first eigenvector, which is E1 here, and if you try to reconstruct your Hi-C matrix from it, it will describe a nice compartmental pattern. So this is original Hi-C matrix, and this is what we can reconstruct from the first eigenvector from compartmental profile, and you can see that it nicely corresponds to the real compartments in the Hi-C. There might be some residual signal that we kind of omit when we analyze compartments, but this is the key, and this vector is usually pretty much bimodal. It's either very negative values, very B compartments, or very positive values, very A active compartments. One small trick here is that the procedure of eigenvector decomposition doesn't know what is A and what is B compartment, and it can flip the sign randomly. And you can see, if we multiply the value by itself, if it's positive, it will produce positive signal. If it's negative, negative multiplied by negative will produce positive signal again. So this procedure is agnostic of the sign. So how do we define A and B compartments after we run eigenvector decomposition? For that, we usually take some additional information about the genome. For example, GC richness, which is associated with active chromatin, with active genes. And then we can try to find those regions that will be positively correlated with GC content and say that that is the positive sign in our eigenvector, and that is what we will call A compartment. It will not change the values of eigenvector, but it will change the sign of it. So, for example, if we first called it not according to our views of A and B compartment, you can see that this value is negative, so it should be considered as B compartment. But, in fact, it is enriched in GC, so it's actually enriched in active genes. That's why we need to flip the sign and then obtain the proper phased eigenvector for the compartments. So, for Hi-C calling of compartments, we need two types of data. We need Hi-C matrix itself, and we need the genome file annotation for annotation of GC content. Let's switch to that part, and let's run in the code. It's section number 3, part 2. And usually for compartments, we load data at the pretty coarse grain resolution, because we need to load all of this matrix in the memory simultaneously. It will be super hard to do at 10 KB or 1 KB, and it also will be noisy. So that's why we use 100 KB usually for compartmental calling. Let's load cooler for this resolution. Next, we download the genome. It might take some time if you do not have it pre-downloaded. And then we calculate GC content. This we do with the bioframe. So bioframe is the library that allows you to do various operations on the genomics data, which is not Hi-C, but maybe information about chromosome sizes or calculating GC content, gene content. So that's where bioframe is really, really handy and comes into the game. So I use this library to calculate GC content. And let's take a look how GC content metrics for a mouse genome will look like. If you are a little bit anxious as me, so you can look at the folder structure that was created during the workshop. So if you press on the left on the folder, you will be able to see what were the data sets that we generated today. So we downloaded the sample data, and you can see 4 files here, and you can see the genome that is currently being downloaded and unpacked. Okay, in my case, the code executed successfully. You might have encountered some troubles. If it was the case, please for now follow the instructions and follow the presentation, and we'll try to figure out what has gone wrong later. So I loaded the FASTA file for the genome, and I calculated GC content. And you can see this nice table where for each 100 kb region, I have annotation of GC content. I will use this data frame later when calculating the compartment profile. So let's progress with that. I will need a simple function of cooltools ig_sys, so it will be sys data used for calculating the compartments. I will input my 100 kb cooler GC content, and I would like to run it for my particular chromosomes, because I know that I do not have the full information for the whole genome, so it is just 2 chromosomes that I work with. Now, let's take a look at the eigenvector track. The calculations might take some time. So what do I mean by cis? What do I mean by taking into account only cis data for eigenvector decomposition? So we can take one chromosome and try to run eigenvector decomposition on it, like I was showing you before. What we can also do, we can take the whole genome information, and we can take interactions between chromosomes, trans interactions, and we try to calculate the same on them. And that will provide to us presumably the same information, because the compartments are also pronounced both in cis and in trans, but in some cases, when compartments might behave differently within and between chromosomes, you might consider one of these options. Okay. So this is the output of cis eigenvector decomposition. For each bin in the genome, we have now E1 value, first eigenvector value. Let's try to visualize it. And here I will take one particular region of the first chromosome, and this code plots for you the matrix and also adds some nice features to the plot that will allow you to visualize the boundaries of your compartments. In this particular case, on the left, lower left will be just untouched matrix, and you can appreciate the checkerboard pattern. And on the right, it will be the result of eigenvector decomposition and finding the boundaries between compartmental switches. And this is the first eigenvector. You can see that it is fluctuating between very negative and very positive values. So these are positive A compartments, negative B compartments. And you can see the boundaries marked on the whole matrix. And indeed, you can appreciate that these blocks are corresponding to the blocks on the Hi-C matrix. Blocks created by the boundaries between domains are corresponding to the blocks in Hi-C matrices. Now, an interesting task will be to do the same, but for RED21 degradation. And here I suggest you to play with the code a little bit. It's more demanding than the previous tasks. So here you actually need to copy some code, and you need to run the same eigenvector decomposition, but for RED21 treated cells. I will do it in parallel, so you will be able to follow or take a look at the result. So what I need is redefine the cooler file. So first, I will need new definition of the cooler file. I will name it 100K RED21. And you can see that the file was reading all right. So it's a proper cooler object. Now I need to run eigendecomposition on it, so I will use this code with a simple cooltools function, and I just need to replace the name of the file with red21. So these are no new eigenvectors for the red21 data. Let's try to visualize it. And here it goes. So just for comparison, I will bring to you the both views between untreated and the red21 treated cells. The answer will be on the right. So on the right, you can see how I calculated it. On the left will be the output of both wild type and red21 treated cells. And what you can see is that compartments are actually quite stable in red21 degradation. And the position of the switches is very similar. And one key difference is that in red21 treated cells, there is less noise, and the compartments look much more smooth. And also, by visual assessment, you probably can see that the matrices are, the compartments are usually more pronounced in red21 treatment. Oh, I actually made a mistake, and I forgot to replace the matrix here with the appropriate one, because you can see this is the code on red21, and I didn't specify it. That's why I was not showing the appropriate matrix associated with high C, or red21 high C. Let's take a look on that. Here it goes. Yes, so that will be appropriate comparison. I apologize for that. So this plot should be actually replaced by this one. And there you can see that the compartments are much more pronounced in red21 treatment. So let's do it again. Here it goes. On the left, wild type. On the right, red21 degradation. And the compartments are indeed more visible. Okay. In the last 15 minutes, 15-20 minutes that we have, let's progress to another typical analysis of high-C data that people do, called scaling plots, or P of S, or sometimes it's also called expected plots. So this is the characteristic that we observed right when we looked at the high-C matrices, that there is very strong main diagonal, and then the strength of interactions is decreasing gradually while we step away from the diagonal. So this property of high C is usually referred to as scaling, contact probability versus distance, and it's a very important characteristic of your high C experiment, which might tell you that you have some troubles with experiment. For example, if you had bad ligation step, that might be reflected on these scaling plots as something unexpected features of your scaling plots. For that, we usually compute the average interactions on the diagonal of the high C matrix. So, for example, here, we will take first diagonal, second diagonal, and further away, calculate their averages and plot them in the simple plot where there is genomic separation on x-axis, so how far we stepped away from the main diagonal, versus average number of interactions. And you can see that this plot is dropping, meaning that we start with very strong interactions at the diagonal, and then it's gradually dropping. Here you can see a very nice example of how that can be interpreted. So basically, we take the perpendicular to the main diagonal, and we see how the average signal is dropping. To run that, again, let's load our cooler data. For this, we can use the highest resolution available for us, 1 KB, which is very fine-grained data. But again, because we will be aggregating it across each diagonal, it will be not that much in the end. So let's try to let's try to use this teeny tiny resolution. This is the basic version of contact versus distance, where we just calculate for each diagonal of high C what is the average interaction. We do this with cooltools expected. might take some time because we selected 1 kb. Here it goes. The output will be information for each chromosome for the particular distance, what is the number of interactions, and you can see them in various forms, like unbalanced or balanced interactions, average or sum, and this will be for both chromosomes. Now let's try to plot this. And notably, when we plot this information, we usually use double logarithmic coordinates, log-log plots, because the drop is such a big change in the strength of interactions that we cannot usually see it well in linear coordinates. It will be just very rapid decay. In logarithmic scale, we usually can see that it's much more perceivable, and we can see that it's a line, more or less. In this case, you can see that there are not really nice features of these plots. They are getting more noisy towards the end of the chromosomes, and they're fluctuating pretty much at the ends of chromosomes. Why so? Because taking diagonals very far from main diagonal will be noisy and sparse, and to deal with that, it's good to have an aggregation. So smoothing and aggregation. So when we will be taking multiple diagonals and averaging signal for all of them, instead of taking one single diagonal. Let's try to run expected with now changed parameter of adding this feature of smoothing and aggregation. And let's try to visualize it. Here it goes. And after smoothing, we have much more nice plot where the large distances are also possible to see and cover. What do we usually do with this information? So we have the scaling plot. We have information that indeed in our data the signal decays with the distance. What's next? So what people usually try to assess is the slope of the scaling plot. So we can try the total distances, the whole distances that we assessed, so this blue line, and we try to take the drop in the contact frequency between most highly interacting and lower interacting regions, and we can try to take these two values, divide them one by another, and get the slope of the scaling plot. So this the slope of this red line, average slope of high C. And that will be informative for us about the properties of general folding of DNA in the nucleus. And again, you can do quality control if it's too flat. So if the slope is not that large, we can be not sure that our high C experiments run well, and might be some trouble with ligation, for example. It might be also the sign of different biology which is happening within our cells. And here, very handy comes the analysis of the derivative of the scaling plot, where basically we do the same procedure as I described here for you for the average slope of high C. But now we repeat that for each little segment of our scaling plot, and for each genomic distance we know what is the local slope there, and we can see that it's indeed not the straight line, and it's some value that is getting more positive and more negative. And that can be a sign of some biological mechanisms acting there. For example, this peak is known to correspond to loop extrusion. So what it means is that there are larger amounts of interactions at the distances of tens to hundreds kilobase pairs. And that is usually the range where cohesin acts. So to test that, let's first try to calculate this derivative, and let's also repeat that for the red 21 data where we know that there is no cohesin. So run the rest of the code in the section 4.4. Perfect, I have it completed. And here it goes. Here is the scaling plot for the wild-type data with the first derivative. And as the next task, I ask you to plot the same, but for red21 degradation. One more tricky thing here is that I include one more treatment of the data that I will tell about later, but let's try to plot both of them at the same time. So here are the different treatments. Blue one is untreated with the nice scaling hump. The orange one is red21 degradation, and now you can see how different this plot is and how flat it is at the distances up to megabase pair and longer. And basically that is the sign of loop extrusion being absent in your data, that there is no more increase of interactions at 100 kb, which is typical distance for the cohesin action. It's all gone in red21 treatment, and that can be nicely visible in your scaling plots. Okay, now about this blue line. We know about players of loop extrusion. We know about cohesin, red21. We know about CTCF, the stop point. But what happens if cohesin proceeds on creating the loops? Usually it falls off with some probability, and there is a special factor that helps to unload cohesin. So this is an example where cohesin was progressing, and then at some point it unloaded. There is no more loop, and cohesin can be reloaded in some other positions in the genome. And this process is facilitated by the factor called wobble. I plotted here in green. And wobble, if we remove wobble, then cohesin doesn't have facilitator of its unloading, so it will be creating larger and larger loops. Indeed, if we take the data for wobble degraded cells, that will create this this green line, basically showing to us that the loops are larger in this particular data set. So with that, there is one more task in the notebook that you might be curious to run. It's related to CTCF and what happens with the scaling plots in the absence of CTCF. Another hidden task here is that, okay, we visualized the scalings for WOPL, but what is in Hi-C for WOPL? And with that, you can play with that during the coffee break and take a look how the Hi-C data looks like for them. And more tips are in this presentation. For example, you can use HiGlass to visualize WOPL data and CTCF degradation data in the ResGen views. I provide here for you the information about these new data sets. Or alternatively, you can browse them on ResGen. So if you follow this particular link, you will be able to browse these data sets by yourself. But generally, I would consider that as advanced tasks. And with that, I would like to conclude with final remarks that I would like really to acknowledge the Mirny Lab, where the software was born and created, and Leonid is my supervisor and mentor. So conference organizers and NSF Physics of Living System program, who was very kind to organize this event and being persistent to do that across multiple years. And the conference supports, Sarah and Louise, who were helping with all the organization, and I really, really appreciate their support. And finally, the enthusiasts who contributed to the software that I was presenting to you today and who meet every week for almost 5 years now to make your experience with Hi-C more smooth and interesting, basically open to see community. Please join our Slack if you are curious, and we will be happy to answer your question. And this is the team and major actors in the Open to See community. So this workshop is largely contributed by them and their efforts. With that, thank you so much, and I look forward to your questions and more discussions during this conference.